Hello friends, I am Shubhangi Rohatgi, PGT Biology from Kendriya Vidyalaya number 1 AFS Hindon, and I welcome you all in the session 3 of chapter 8 Human Health and Disease from class 12th unit 8. In the earlier session on this chapter, we had studied in detail the human immune system and we had discussed the various components of this immune system and also how they work together to ward off the infections. In the last part of the class, I had given you some questions. Actually, it was a list of some related terms and you were to differentiate between them. I hope that you have tried to differentiate between them. Let us have a quick look on the answers of some of them. Let us begin with the first pair, which was primary and secondary lymphoid organs. As we had discussed in the class that primary lymphoid organs are those where immature lymphocytes differentiate, while the secondary lymphoid organs are those which provide the site for interaction of lymphocytes with the antigens. Another point of difference that you can add here is the example. You can mention that examples of primary lymphoid organs are bone marrow and thymus, while those of secondary lymphoid organs are spleen, lymph nodes, tonsils and Peyer's patches. Next up the list was differences between innate immunity and acquired immunity. Again, this was a very easy pair, as we know that innate immunity is present in us since our birth, while acquired immunity is the one which develops during our lifetime. The innate immunity is mainly conferred by various barriers like physiological, physical or cytokine barriers. On the other hand, acquired immunity is given by mainly lymphocytes like B and T cells. Another important point of difference is that in innate immunity we say that it is a non-specific kind of defense system, that is, it is not specific towards any pathogen. On the other hand, in acquired immunity we see that it is pathogen specific, it is highly specific. Furthermore, it shows the property of memory, that is, if it encounters a pathogen for the first time, it remembers it, and whenever that very pathogen attacks another time, then it responds with much more vigor. So, acquired immunity is different from innate immunity in all these aspects, especially being pathogen specific and having the memory. The next pair was somewhat tricky and it was of cytokines and lysozymes. Well, we see that cytokines and lysozymes are similar in the respect that they both form the part of innate immune system, but then how are they different? Well, again they are different because cytokines are produced mostly by cells like leukocytes. On the other hand, lysozymes are enzymes which are present in tears, saliva, mucus, etc. Furthermore, we know that cytokines mainly act against the viral infections, while the lysozymes are there for the bacteria. They degrade the cell wall of the bacteria, thereby killing them. Another pair on the list was allergies and autoimmune diseases. Again, both of these terms and conditions appear to be similar. How? Because in both these cases, the defense system seems to be going haywire. It is acting against the molecules or compounds which are otherwise known to be harmless, and the defense system should not be actually acting against them. But the difference lies in the fact that in case of allergies, the reactions are against the foreign antigens, while in case of immune diseases, autoimmune diseases, reactions are against the self antigens, and hence these reactions are called the source of autoimmune diseases. So, in case of allergies, the immune system starts acting against some foreign origin objects like pollen grains or other things, maybe in lactose, which should or would have been otherwise harmless, totally harmless, but the immune system is acting against them. On the other hand, in case of autoimmune diseases, what is happening? The immune system is killing the body cells of its own. So, in this way they are two different. Now, this was all about the last class. Let us move on towards the today's topics. Today we are going to discuss two very dreadful diseases. One is AIDS and other is cancer. AIDS, as we know that, stands for Acquired Immunodeficiency Syndrome. What is the meaning of this term? Well, this explains actually what happens in AIDS to the body. Let us see. AIDS means it is a condition, a group of system which arises when there is deficiency of immunity in the body, and this deficiency of immunity is something which is acquired. It was not innate, it was not with which the person was born. It has been acquired deficiency of immunity, and it is leading to a group of symptoms which consistently occur together. So, this is acquired immunodeficiency syndrome. Let us take a look on the brief history of AIDS. It is believed that about 100 years ago there existed a SIV, simian immunodeficiency virus. It was present in chimpanzees. Now, what must have happened is that this SIV got transmitted to humans when humans hunted chimpanzees for meat and came in contact with their infected blood. Afterwards, the HIV after entering the humans, SIV after entering the humans, it got mutated into HIV. It is a presumption. The earliest known case of AIDS infection was reported in 1959, and in over the coming 20 years, we see that AIDS became a global pandemic. It had infected so many people by early 1980s that it was declared to be a pandemic. It was only in 1982 that the term AIDS was coined, and it was used to describe the occurrence of opportunistic infections like Kaposi's sarcoma and pneumocystis pneumonia in previously healthy people. A year later, the causal organism, the pathogen responsible for this condition AIDS, was discovered, and it was called HTLV3 or LAV. Today we know this virus by the name of HIV, and by 2015 this deadly virus HIV had infected more than 70 million people all over the globe, and out of those 70 million people, about 50% had died. So, we can see here that this virus has spread with an alarming rate. There must be something in the way in which it spreads. That is why it is infecting so many people in such a short span of time. So, let us have a look on the ways by which this virus spreads. You all must have heard that HIV spreads through the body fluids like blood, semen, vaginal fluids or the mother's milk. So, the common routes of transmission of AIDS include blood transfusions or sharing of infected needles, unprotected physical contact, or from mother to the baby through placenta or through the breast milk. But have you ever thought, are these the only body fluids that are present in our body? Are not there other fluids which can transmit HIV? What about saliva? Can saliva transmit AIDS? Let us see. Well, if a person is HIV positive, his or her saliva will definitely carry the HIV because it is present in his or her system. But the saliva has many other compounds also, which makes it impossible for the HIV to infect other cells. There are many compounds which are inhibitory in this respect. There are HIV specific antibodies which neutralize and inactivate the virus. Then there are cystatins and defensins which are generally antimicrobial against the viruses. There are lactoperoxidases, mucins, secretory leukocyte protease inhibitors and thrombospondins. All of these act together in order to prevent the HIV which is present in the saliva from infecting the other cells. Why does not AIDS spread through casual contacts then? Okay, so we have known that it is present in the saliva, but because of these inhibitory compounds, it cannot infect the other cells when present in saliva. But there are so many viruses which get spread only through casual contacts, or we can say indirect contact. Take for example the flu virus, which spreads so rapidly through indirect contact of objects which have been touched by the infected person. What about AIDS? Does AIDS spread in this manner? No, it does not. We know this for sure. But why? Why it so happens that AIDS cannot spread through indirect contact? Any answers? Well, yes, you are right if you are thinking that HIV virus cannot survive outside the cells. That is why it does not spread through the contact of objects used by an infected person. Are there other routes of transmission also which we can discuss here? What about mosquito bites? Can HIV spread through insect bite? Why or why not? I ask you to think of any instance. Have you ever heard of any instance where you have come to know that HIV spread through mosquito bites? Yes, you are right. You must not have heard of any such instance, and that is because the HIV cannot survive even inside the insects. Okay? So, HIV basically survives only in our cells. Let us study the structure of HIV in order to understand its life cycle in a little bit more detail. The HIV can be divided into three parts: the genome, the capsid and the envelope. The genome is RNA genome. It is a retrovirus, we know that. It has 2 copies of single stranded RNA and which code for the virus's 9 genes. These 9 genes code for the proteins which are responsible for its replication and infection. Outside of genome, we find there is capsid. The capsid is composed of viral proteins, and the single stranded RNA which was present in the genome is tightly bound to the nucleocapsid proteins, and it also contains the enzymes which are needed for the development of the virus. The outermost layer is the envelope of the virus, and you will be surprised to know that it is composed of the lipid bilayer taken from the human cell. Also, it contains some of the human proteins, but it also has some proteins which are coded by the virus, and these are the proteins which are coded by viral ENV gene, and they help the virus in attaching to the target cell and also in fusing to the target cell. Let us have a look on the structure of HIV virus. So, if this is the virus, this here is the genome made up of 2 single stranded RNAs. It also contains the enzymes which are required by the virus for completion of its life cycle. This is the capsid. Outside the capsid is the envelope. This envelope is made up of lipid bilayer taken from the human cells, and on this there are envelope proteins. Some of the envelope proteins which are coded by the ENV gene of the virus are helpful in attaching the virus particles to the target cells and also in fusing through the target cells. Moving on, let us see how the HIV replicates inside the human cells. Here, this diagram is given in your NCERT textbooks. Let us study this diagram in details. So, here we can see there is the viral particle with its RNA genome. The virus particle infects the normal cell and the viral RNA is released inside the cytoplasm. What happens next? This RNA is used to make DNA by the process of reverse transcription. Now, one question here. Okay, the RNA can give rise to DNA by reverse transcription, but not on its own. It must require some cellular machinery. It must be requiring some enzymes, some other proteins which help it to form the DNA. From where does this machinery for reverse transcription come in the cell. What do you think? Should it be present in the host cell itself, so that whenever a retrovirus attacks, the machinery that was present in the host cell can easily be used by the retrovirus for making its own DNA? No, of course not. The machinery, especially the enzyme, the reverse transcriptase enzyme, is carried with the virus only inside the nucleocapsid region. Here I will give reference to the diagram which I had shown earlier. We had shown the enzymes here. These are the enzymes which are crucial for the replication and infection process. So, reverse transcriptase is present in the nucleocapsid region, and it is used for reverse transcription, and the viral DNA is formed. Now, viral DNA gets incorporated into the host genome. Again, for incorporation, enzymes like integrase will be used, and again this integrase enzyme is produced by the virus itself. Once the viral DNA has got incorporated into the normal host DNA, what will happen? When it will be transcribed, viral RNA will be produced, and this viral RNA will be used to make many more viral particles inside the cell. When the cell will burst, these viral particles will be released. This was the replication cycle of the HIV in human cells. But can you see the immune system anywhere in the picture? So, how do we say that these viruses affect the immune system? Let us see how. Actually, it so happens that when HIV gains entry in the body, it enters the macrophages, and firstly it replicates inside the macrophages, and when new viral particles are released from the macrophages, they enter the helper T lymphocytes. Now, these T lymphocytes are used to make new viral particles and infect more and more T cells. What will happen? The T cells also burst in the end. As a result, the number of T cells decreases. So, can you see what is happening here? The cellular army that was responsible for our protection against various pathogens is now being compromised. Not only the T cells get killed in the process, but before being killed, they are being used as a factory for manufacturing the viruses. So, instead of giving us the protection, they are producing more and more viral particles. As a result, the number of viral particles increases, and in the same proportion the number of T cells will decreases. These T cells are the cells which are responsible for identification of the antigen, for their processing, for helping the B cells to produce the antibodies against the antigens. Once their number gets low, what will happen? All the other microbes which were waiting for such an opportunity to attack our cells will now enter the body. Your defense, your line of defense has been compromised. So, all the microbes which would otherwise not be able to infect your body will now get a chance to enter and attack. So, there will be a host of infections in the body now, and these infections are known as opportunistic infections. And most of the times, the person who is suffering with AIDS dies of these opportunistic infections. But all these series of events, they do not occur overnight or in a few days. In fact, the HIV infection and its progression to AIDS takes years. Let us see how. So, there are stages of HIV infection. The first stage is acute HIV infection stage, followed by clinical latency stage or chronic infection stage, and finally the AIDS. Let us see what happens in each of these stages. During the acute infection stage, what happens? The flu-like symptoms appear in the initial weeks. When the infection has just occurred, the symptoms of flu will appear, and people have been reported to say that it is the worst flu ever. Actually, what is happening inside the body? The body is trying to ward off the HIV which has gained the entry. So, all these flu-like symptoms are the response of the body for getting rid, when it is trying to getting rid of the HIV, but unfortunately it fails to do so. So, what happens? The large amounts of viruses are being produced in the body during this stage. The viruses use CD4 cell or the Th cells, helper T cells, to replicate and destroys them in the process, and because of this the CD4 count falls rapidly. Here you can see in this diagram, if earlier CD4 count was this, it has reduced to this much. We can see this with the help of graph also. The blue line represents the number of Th cells, that is the CD4 cells, and the green line shows the viral load. So, we can see here in the acute infection stage, the viral load increases and CD4 cells decrease in number very sharply. After this comes the clinical latency stage. What happens here? The people experience no symptoms, or if they are present, they are only the mild ones. What is happening inside the body? Actually, at this stage there has been achieved a balance between the infectious HIV and the CD4 cells. What is happening? The HIV are still replicating, but at very low stage. The T4 cells are still at a reduced amount, but still the body is able to sustain itself. So, both CD4 cells and the HIV virus are present there in the clinical latency stage. The person is asymptomatic, and at this time if ARTs are given, this stage can be made to last very long, almost up to the normal lifespan of a normal human being. I have a question for you at this stage. Now, we have discussed that at the chronic infection stage the HIV is not replicating and the person does not show any symptoms at all, or even if the symptoms are there, they are very mild. Person does not really seem to be very ill. So, such a person who has a chronic infection at this stage, will he or she be still transmitting HIV through various routes? Yes or no? Of course, yes, because the HIV virus is still present inside his or her body. So, what that it is replicating at somewhat lower rates, but still it is active. If antiretroviral therapy is not given at this stage, what will happen? The chronic infection stage will progress towards AIDS, and this chronic infection stage lasts for up to 10 years on an average. So, what happens when AIDS is detected, or when do we say it is AIDS? When the CD4 cell count has decreased to very low levels. In normal human beings, it is about 500 to 1600 cells per millimeter cube of the blood, while when there is AIDS, it is reduced to below 200 cells per millimeter cube of the blood. At this stage, what is happening? The viral particles increase in number and the CD4 count has decreased. So, what will happen? The opportunistic infections will now occur and they will make the body very sick. And as I said earlier, many people die of these opportunistic infections. Without treatment, people who progress to AIDS typically survive about 3 years. So, we now know that the virus spreads very rapidly and very rapidly it infects the cells of the immune system. So, we need to develop some techniques for early diagnosis of the virus in the system, so that ARTs, the antiretroviral therapy, can be started in time. And that tool, one of those tools that are used for its diagnosis are ELISA. ELISA can be used in fact to detect any viral infection, any virus. This test takes advantage of the interactions between antigens and the antibodies. The full form of ELISA is enzyme linked immunosorbent assay, and if this is positive, a Western blot test is usually administered to confirm the diagnosis. Let us see how ELISA helps to detect the HIV infection. What is done? Wells are made and in these wells antibodies are made to bind. sometimes we can go other way around also, that we take an antigen and bind it to the bottom of the well, but here in this diagram we are working with the antibody. So, one antibody specific to the antigen that is being studied is made to bind at the bottom of the well. After several washing offs, the sample which is to be studied is added. If the antigen is present in the sample, what will happen? It will bind to the antibody. Now, another antibody is added, but this time this antibody is compounded with a chromogenic compound, and this antibody again will target the same antigen and it will bind the antigen, and because of this binding a color will be produced. On the other hand, if there was no antigen to begin with, so there will be no binding at the first step, no binding at the second step either, and hence no color formation. So, simply by looking at the colors of the test tubes we can know whether the antigen is present in the sample or not. But there is a problem. During the initial stages, that is known as the window period, the amount of antibodies produced against the virus are very low, which cannot be detected by this ELISA test. Okay. So, ELISA otherwise is quite effective test, but it does not work in the initial period, that is known as the window period. Talking about the treatments of the AIDS, we all know that human body can't get rid of HIV completely even with the treatment. So, once HIV has gotten, the infection has set and the person has to live with it. It is just like many other diseases like diabetes. We cannot get rid of these diseases totally, but yet with medicines, with proper care, we can make the person live a normal life, near normal life. And these ARTs, which are the antiretroviral therapy, they target to control the HIV. They cannot kill the HIV, but they target to control the HIV, they prevent its multiplication. If ARTs are not given, HIV will destroy the immune system completely and the person will progress to AIDS eventually, but if ARTs are given, then AIDS can be delayed. So, the HIV medicines that are being used in antiretroviral therapy, they basically target to inhibit the replication, the infection or the release of the viral particles in the bloodstream. So, there are reverse transcriptase inhibitors, there are protease inhibitors, fusion inhibitors, entry inhibitors and strand transfer inhibitors. All these drugs work with a single name, that is to prevent the virus from infecting new cells and replicating or making its many copies inside the cells. So, there is no treatment. What should then be done? We should move to prevention. Government of India has been running many programs to educate people about AIDS and help them to prevent or stay away from AIDS. I think knowing the routes through which the AIDS spreads can help us to prevent the disease. How? By simply avoiding those routes. If we avoid the routes of transmission or routes of spread, then we can safely protect us and our loved beings from getting the deadly disease AIDS. This was all about AIDS. Now, let us move on to cancer. Cancer is again one of the very dreadful diseases. What actually is cancer? Cancer is a class of diseases characterized by out of control cell growth. What happens? Cancer harms the body when the altered cells divide uncontrollably to form lumps or masses of tissues called tumors. And these tumors can grow and interfere with the different systems of the body like digestive system, nervous system, circulatory system, etc. And they can also release hormones that alter the body function. Normally, human cells grow and divide to form new cells as the body needs them. When cells grow old or become damaged, they die and new cells take their place. But when cancer develops, this orderly process breaks down. As cells become more and more abnormal, what starts happening? Old or damaged cells survive when they should have died, and new cells form when they are not needed. These extra cells can divide without stopping and may form growth called tumors. Now, let us look here for an example. In case of a multicellular organism, all the cells seem to live in a harmony. They grow when they are needed to grow, and they divide when they are required to divide. They divide when some repair work has to be done, some damage has been done and it has to be reversed, then only they divide. Otherwise, they stop dividing. They only divide when it is required of them to do so. What would happen, suppose if in an organ some cells go haywire, they stop obeying the rules of this cell division, and instead they start dividing rapidly without any control. What will happen? A lot of new cells will be produced, and these new cells will appear as a lump or mass of cells. This mass of cell is known as tumor, and these tumors interfere with the normal functioning of the body. This is cancer. Tumors can be of different types, benign and malignant. The benign tumors are those which remain confined to their place of origin, while the malignant tumors invade and damage the nearby tissues. So, which one would be more dangerous? Obviously, benign tumors are comparatively easier to remove by surgery. On the other hand, it is very difficult to remove the malignant tumors because they show the property of metastasis, that is, they can move from their place of origin to a newer part of the body and produce a new tumor there. So, within few months the cancer spreads all through the body. So, that is why malignant tumors are more difficult to control. Let us talk about the metastasis. What happens? A cancer that has spread from the place where it first started to another place in the body is called metastatic cancer. What is happening here? In metastasis, cancer cells break away from where they first formed, that is the primary cancer, and travel through the blood or lymph system and form new tumors. These are known as metastatic tumors in other parts of the body. Obviously, the metastatic cancer, that is the secondary cancer, has the same name and same type of cancer cells as the original or primary cancer. For example, breast cancer that spreads to and forms a metastatic tumor in the lung is a metastatic breast cancer and not a lung cancer. Metastatic tumors can cause severe damage to the body functions, and most people who die of cancer die of metastatic disease. Let us try to compare the cancer cells with the normal cells, so that we can comprehend in a better way what makes cancer cells what they are, what makes them to go haywire and divide uncontrollably. First thing is that they do not show the property of contact inhibition. Now, what happens in normal cells, they have this property of contact inhibition, whereby by virtue of which these normal cells, when they come in contact with the other neighboring cells, they stop dividing. This is a cue that since they are in touch with the other cells, there is no place to produce new cells, so do not produce new cells, and they take this cue. On the other hand, cancerous cells, they do not show this property. Even if they are in contact with other cells, they will keep on dividing. The next difference that is present between cancerous cells and normal cells is that the cancerous cells are less specialized than the normal cells. This is one reason that unlike normal cells, cancer cells continue to divide without stopping. What happens is that normal cells have differentiated, they have obtained their structure and they are performing their normal functions. So, they do not actually have time and resources and need for division. On the other hand, the cancerous cells are yet to be specialized, they are usually not differentiated, they are very less specialized, so they can continue to divide. The other difference that makes cancer cells different from normal cells is that they are able to ignore the signals that normally tells the cells to stop dividing or that begin programmed cell death or apoptosis. What is apoptosis? See, apoptosis is a mechanism by which body can get rid of unneeded cells. There are many times when body needs to get rid of some cells. Can you think of some times when this must happen? Don't get confused in thinking that growth only involves production of new cells. It is not always that, especially if we talk about development. Development not always means increase in number of cells. It can also mean decrease of number of cells, and apoptosis plays a very important role there. Think about embryonic development when the digits are needed to be made. Initially all the digits are united, but then they are to be carved out and made separate, and the cells between these digits are to be removed, and these cells are removed by apoptosis. There are many other examples also where body gets rid of unwanted cells through apoptosis, and so that they do not divide actually. And the cancerous cells, they ignore the cues for apoptosis, they ignore the cues for programmed cell death, and instead they continuously keep on dividing. Also, cancer cells influence the normal cells and molecules, blood vessels that surround the cancer cell and feed the tumor. Finally, they are able to evade the immune system. Immune system has this ability that it can sense that something is going wrong and it can try to remove the cancerous cells, but here the cancer cells seem to be smarter, they are able to evade the immune system. Also, cancerous cells have more mutations in their DNA than the normal cells. Now, one thing is to be noted here, that cancer actually arises because of mutations in the DNA. That is one thing, but since these cancer cells are continuously dividing at such fast rates, and they are dividing and undergoing DNA replication at such great rates, that mutations continue to accumulate in these cancerous cells. So, more often than not, the mutations that we see inside the cancerous cells are the results of their being the cancer cells, not the cause of them being the cancer. Okay. So, this is how the cancer cells are different from the normal cells. Let us have a look on the types of cancers. Actually, the types of cancers, there are different types of cancers and they are named according to the cells from which they originated initially. For example, carcinomas are the cancer of the epithelial cells of any tissue in the body. Similarly, sarcoma are the cancers of connective tissue, leukemia of the bone marrow, lymphoma is the name given to cancer of lymphocytes, so on and so forth. So, there can be various different cells which can take part in cancer, which can cause cancer, and accordingly there is different nomenclature. But the question is how cancer arises. As I had said earlier, that at the root cause of the cancer is mutation. The mutations are taking place in the DNA, and because of these mutations the DNA starts sending wrong information, the cell division stops, the cell division fails to stop, and the cells keep on dividing continuously. So, how do these mutations affect the normal cells? What happens when the mutations occur? These mutations cause the cell to stop its normal function and allow a cell to become cancerous. Now, each person's cancer has a unique combination of mutations, not two mutations are same generally. As the cancer continues to grow, additional changes will occur, that is more and more mutations will accumulate. I had just said that even within the same tumor, different cells may have different genetic changes or different mutations. So, what are the causes of cancer? They are the gene mutations, but where are they arising or how are they arising? lets us to differentiate between them. So, hereditary cancers include the proto-oncogenes and DNA repair genes, while acquired causes include chemical, biological and physiological carcinogens. Let us have a look at each of them. Talking about the genes, there are these, let us have a look at these three genes: proto-oncogenes, tumor suppressor genes and DNA repair genes. All these three genes are involved in normal cell growth and division. DNA repair genes are involved in fixing the damaged DNA. What will happen if any kind of mutation takes place in these genes? These genes are very important and crucial for normal cell growth and division. They are controlling the DNA repair function also. If any kind of mutation takes place inside these genes, these systems will start functioning wrongly. So, what happens if these genes are mutated? They allow the rapid growth of the cell, then they fail to stop the uncontrolled cell growth, and they make mistakes when repairing the DNA errors. Cells with these mutations in these genes tend to develop additional mutations in other genes as well, and all these can lead to formation of cancers. Talking about the carcinogens, chemical carcinogens, there are several chemicals and environmental toxins which are responsible for changes in normal cellular DNA. Such substances are known as mutagens, and if these mutations are cancerous, then these chemicals will be known as carcinogens. Some of the commonest carcinogens are tobacco, alcohol, asbestos fibers. Now, tobacco not only causes lung cancer, but it is related to many other cancers like that of lungs, larynx, head, neck, stomach, bladder, kidney, esophagus and even pancreas, because it has many other carcinogens like nitrosamines and polycyclic aromatic hydrocarbons. Even our many day-to-day items like dyes, paints, air fresheners, antiperspirants also carry a host of carcinogens. So, how is this information helpful for you? You better check the labels of different chemicals and products that you use in your day-to-day life, and look for anything that says it is known to show carcinogenic properties. Better safe. The next reason for causing cancers is ionizing radiations. So, we know of ultraviolet rays, that ultraviolet rays from the sun can lead to melanoma and other skin malignancies. What about radon gas? It says here that the exposure to this gas can also cause cancer, but do you know of anything that emits radon gas and we are in its exposure in our day-to-day life? Yes, it is the granite stone. The granite stone, the slabs which are used in making the kitchen and our household areas or offices, this granite stone is known to produce radon gas, and radon gas can cause cancers. Unfortunately, radiation therapy given for one type of cancer may also cause another type of cancer. This is really an unfortunate instance. Well, there are other causes also. Firstly, there are viral and bacterial infections. Many viral and bacterial infections are known to cause cancers. For example, liver cancers because of hepatitis B and C infections, then cervical cancer due to human papilloma virus, and Burkitt's lymphoma because of Epstein-Barr virus, and gastric cancer because of Helicobacter pylori infection. So, these viruses and bacteria, when they enter the cells, they manipulate our genome, manipulate our genes in such a manner that we get cancers. There are genetic or inherited cancers also, that is, these are the genes which when mutated give cancer, and these are these mutations are inheritable. Example, inherited breast cancer and ovarian cancer because of genes BRCA1 and 2. These genes have been in news lately. Then there is Lee-Fraumeni syndrome, which includes defects in P53 gene that leads to many types of cancers like bone cancers, breast cancers, soft tissue sarcomas and brain cancers. Even Down syndrome, it is known to develop many abnormalities like leukemia and testicular cancer. How is cancer detected? It is important because early detection and diagnosis is very imperative and plays a very important role in curing or treating the cancer and prolonging the life of the cancer patient. There are various methods. Firstly, there are lab tests. The lab tests include the test of chemical components like glucose, electrolytes, etc., and even the amount of oxygen and proteins in the blood. Then there can be urine tests to check the blood, chemicals, bacteria and other cells for infection or other abnormalities. Then there are diagnostic imaging processes. 3 types of imaging processes are used to visualize the cells and tissues and organs of the body inside. These are transmission imaging, reflection imaging and emission imaging. Let us study them. Firstly, transmission imaging. In this method, a beam of high energy protons is passed through the body structure being examined. What happens when there are less dense tissues, soft tissues, watery secretions, blood and fat, etc., the beam passes through them very quickly, leaving a darkened area on the X-ray film. However, the more denser tissues appear to be grey and most dense areas appear whitish. So, that is why the bones will appear white, muscles and connective tissues will appear grey and softer areas will appear blackish on the film. Some radiological examinations where images are produced by this method are, we know of X-rays, then CT scans, fluoroscopy, bone scans, lymphangiograms and mammograms. These methods make use of the process of transmission imaging. Then there is reflection imaging, where reflections are images are produced by sending high frequency sounds to the body part or the organ being studied. These sound waves bounce off of the various types of body tissues and structures at varying speeds depending on the density of the tissue present. The bounced sound waves are sent to a computer that analyzes the sound waves and produces a visual image of the body part or the structure. You must be knowing how ultrasound works. So, the sound waves are made to hit the organ or the structure being studied, and from there they get bounced off. They are collected here by the computer, and when we study the frequency and the timings for timings taken by the sound waves to reach the analyzer, here we can get a picture of the structure. This is how the ultrasound works. Moving on to the emission imaging. Emission imaging occurs when tiny nuclear particles or magnetic energy are detected by a scanner and analyzed by computer to produce an image of the structure or organ being examined. Take for an example MRI. The MRI uses radio waves with a machine that creates a strong magnetic field that in turn causes cells to emit their own radio frequencies. A chemical named gadolinium is injected into the veins while performing the MRI. What happens? This gadolinium, it gets collected around the cancer cells, so they show up brighter in the picture. MRI is a very useful tool. It can be used instead of a CT scans in situations where organs or soft tissues are being studied, because with MRI scanning, bones do not obscure the images of organs and soft tissues as does CT scanning. Okay. So, MRI can be used for soft tissues because bones do not create an obstruction while taking the images. Moving on, there are other methods also. Endoscopic examinations. What do we mean by endoscopic examinations? Well, there is a tube with a camera, tiny camera attached to one end. These tubes are sent inside the body through the natural openings like mouth, and then the camera takes pictures of the internal organs and structures. So, the cancers or the tumors can be directly seen by the doctor who is studying the or who is carrying out the endoscopy. Depending depending upon the different organs of which endoscopy is being done, there are different names for different procedures. Moving on, direct DNA studies can also be utilized to find out the cancer. How? Let us see. DNA of the cancer patient is screened for mutation. If we know that a particular person has cancer, his or her DNA will be studied and mutations will be tried to be found out. If mutations associated with cancer are detected, it will be good, and then these sequences of DNA which carry these mutations are radio labeled, and a radio labeled probe is made with this mutated gene. Now, this radio labeled probe can now be used to detect whether the same mutated gene is present in the relatives of that cancer patient or not. So, what will be done? Hybridization with DNA to be tested will be done, and if hybridization occurs, we will see under the autoradiography, we can see whether the hybridization with the radio labeled probe has occurred or not. If hybridization occurs, that means the person in question is susceptible to the same cancer. However, if hybridization does not occur, it can be safely said that this mutation for this particular cancer does not exist in that person. So, these type of techniques are very useful in determining whether a person can get cancer in future or not, if his or her relative has that cancer. Next up the list is indirect DNA studies. Indirect DNA studies are done where the gene that is associated with the cancer is very difficult to be isolated or studied. In that case, we make use of another segment of the gene DNA, which is closely linked with the gene in question, the gene which causes the cancer. So, this suppose this is the stretch of DNA and this is gene or the mutation associated with cancer, we cannot isolate it somehow. So, we look for a marker here, we look for a region which appears to be closely linked with the gene. That means, if the marker is present, it can be safely presumed that this particular gene will also be present. So, instead of looking for the gene, we look for the markers. These are the indirect DNA studies and can be used to detect cancers. Antibodies can also be used to detect cancers, and then we have tumor biopsies also. What are tumor biopsies? The doctor or the surgeon takes out the tumor cells and studies them under the microscope. Then, talking about the treatment, the common treatments are chemotherapy, radiation therapy, surgery and biological therapies. But the problem with all these therapies is that they have their own set of side effects. Chemotherapy makes use of drugs that are that can kill the cancer cells, and radiation therapy, it makes use of high energy radiations to kill or shrink the cancer cells. But these therapies can also damage the normal cells, which can lead to side effects. Immunotherapy, we had said earlier that immune system can help to kill the cancer cell. In immune system, the body's immune system is given a boost in order to be able to fight the cancers. Talking about surgeries, surgeries can help in many ways to remove the tumor, to diagnose the tumor, to study the stage or support other cancer treatments also. There are other treatments also like hormone therapy, blood and bone marrow transplant, biological therapies and angiogenesis inhibitors. All these therapies aim to stop the growth of the cancer cells by use of hormones, transplants or biological compounds, and in angiogenesis inhibitors we use inhibitors to prevent the growth of new blood vessels. This was all about cancer. Now, I would like you to have a look on these questions and try to find answers to these questions. I will read them for you. Please find an example which supports the presumption of RNA world. Analyze the difference between being HIV positive and having AIDS, and Also, can you infer which barriers of immunity are impervious to the HIV while others are easily broken? Which ones are easily broken? How do ARDs help HIV infected persons to lead a normal life? Please try to frame the answers from the studies that we have done. What role do alpha interferons play in treatment of cancer? Why do cancer cells of immune system fail to eliminate cancer? What is neoplasticity? How is it harmful? How can cancer be avoided? And why is it so important to spread awareness amongst the teenager about cancer and AIDS? I will stop here. Thank you.